Good afternoon, and I welcome you all to the 15th SACS Forum, to the Rising Star Seed session. Unfortunately, we are still in the COVID period. That's why we are doing it this way. Hopefully, in the next SACS session, we'll all be able to meet in person, one way or the other. So as your moderator, I will not spend much time other than to say that I'm very happy to see the new companies that are presenting today. They have a diverse portfolio of things in software, small molecules, antibodies. And as you all know that you are the rising stars of tomorrow. So this is your forum. And as we indicated, I hope you will be able to make your case very well and get what you desire to get from a SACS conference. And I wish you all good luck. And before I give you individually a chance, I would like to introduce Martyna from BaseLaunch. And Martyna will tell you how an organization like Basel Innovation Center can actually support you if you ever need it. And that's a great way, and they have a fantastic track record. Not that because I'm from Basel that I'm saying this, but it's a fact. You all know it. And so, Martyna, please tell how you can help all these people whenever they need help.
Good afternoon, everyone. Thank you very much for the invitation. My name is Martyna Wroblewska. I'm a manager of innovation and sourcing at BaseLaunch. And today I would like to tell you about our experience with incubating very early stage biotechs. So at BaseLaunch, we partner with scientists and entrepreneurs, and we help them to launch and grow really exceptional biotech ventures that are developing cutting-edge therapeutics. And we know how to do this because many of our portfolio companies, they have had great successes. And here we can define a success in a different way. So for example, we can look at the amount of money that our companies have collectively raised. And at the moment, this is over $390 million. Or we can have a look at closed partnerships or our companies being acquired. So now I would like to focus on what are the early steps to build a successful biotech venture. And first of all, let me point out that, of course, having a successful biotech company really requires to have exceptional and very solid science. But since this topic is not science-based, I would like to focus on everything that's supposed to surround this science. So building and growing biotech company really needs a very clearly defined path that can, help you to sequentially take out the risk. And you also would need a very well articulated strategy that you really have to follow. And of course, you would also need a team to deliver it. And team is actually very important because really biotech requires lots of talent and lots of experience. And of course, in some places in the world, it's a little bit easier to come across this talent and experience than in the others. But here in Switzerland, we are of course very lucky because Switzerland is renowned by high quality of life, reasonable taxes, and also a very well-known biotech cluster, which of course is Basel region. But Switzerland itself is really an exceptional place, a really great place to do the research. So Switzerland is either among the leading centers in biotech or is really catching up very quickly. If we have a look here at this graph, then we can learn that when it comes to different modalities, then Switzerland is really leading in antibody development and catching up very quickly in cell and gene therapy and immunotherapies, as well as services. And when it comes to different therapeutic areas, then Switzerland is leading in central nervous system diseases, dermatology, and growing very rapidly in oncology. So it's a really great place to do your science. And of course, successful biotechnology also rests on this very supportive ecosystem, and this is really to facilitate this exchange of collaborative efforts and exchange of knowledge. And of course, it also requires the connectivity with larger pharma and biotech companies. And this is to ensure this two-way talent flow when you can ensure that you would have appropriate experience. For example, you can have some entrepreneurs in residence joining your company, but also it can help you to facilitate partnerships as well as to facilitate in and out licensing. But of course, large pharma and biotech alone are not enough. You really need a cluster. And here we have a great example of what a proper biotech cluster really is. So it's not only a few big companies, but also abundance of mid-sized companies and plenty of startups that really helps to facilitate all of this collaborative effort. So Basel is really one of Europe's leading life science hubs with over 700 companies located here and over 1,000 research groups. And it is also a very well-founded region. So Basel region-based companies raised over $6 billion in financing rounds in the last few years. So this should be definitely encouraging to anyone who would like to set up a company here. So at BaseLaunch, we source pharma ventures from all across Europe. Since 2018, we screened over 800 projects, and at the moment, we focus on therapeutics only. But within therapeutics, we go very broadly, so we consider all across disease areas and different modalities. Of course, as long as projects are really innovative, they're backed by solid science and realistic route to Series A funding. So now just a few words about what really early stage biotechs need and how we can support them at BaseLaunch. So first of all, we think that having appropriately built team is definitely the most important thing. So we will be working with you to bring in this drug development and related expertise. And so having this biopharma experience is really crucial, of course, along with access to capital and high quality of science. But of course, it's also important to realize that when you're just at the beginning of your journey, it is very difficult to attract this senior expertise, and that's why we will be working with you to help to establish those fair ways to compensate your early team members. Here, of course, we can think that one could offer participation in the ownership, and here we could also consider that, for example, equity could be a good vehicle to recruit such senior team members. In addition to that, we will be also very heavily focused on providing support with strengthening your IP strategy and setting up the foundations of your company correctly. This is, of course, very important because any mistakes in this field can make your company potentially unfundable in the future. So having all of these steps done right can help you and can save you a lot of headache in the future. So it is also important to realize very early on that IP is your most valuable asset, and therefore you should work with us, and we should work together to make sure that your technology is very broadly protected, as broadly as possible. And of course, we'll also support you to seek early counsel when licensing IP from a third party. So in addition to that, we will be also working to help with this strategic support to really position your company when it comes to different choosing appropriate therapeutic area, also indication, to considering your competition, and also supporting with business development and more. And we will also help to define those key scientific milestones and to further de-risk your science. So in addition to that, we will be also linking you globally to venture funds and assisting in negotiation with these funds. So of course, it's also important to realize that many VCs will typically invest a little bit later in the pipeline compared to incubators in accelerators, but nevertheless, it is very important to build a relationship with those investors early on. And here, if you have a look at this graph, it's just an example that investment in European biotech has more than doubled in the last few years. So there is definitely lots of appetite from the investor side. But of course, you will be also needing a very early financial support to really get your company going, to help you to reach those critical milestones and to answer those go and no-go decisions. And that's why at BaseLaunch, we can help you to provide up to $500,000 in financing. This is in form of convertible loan, as well as access to our partners. So we are backed by 7 partners that you can see over here. These are 6 biopharma companies and 1 venture capital. And to the companies that are working with us, we can offer access, strong access to their know-how, capabilities, and of course, additional equity investment. So we will be also focusing together and working on your company building and also on your team building. So we will support you to bring this drug development expertise, and we can also offer access to our labs and infrastructures. So we collaborate with 2 technology parks. One is Switzerland Innovation Park Basel area, and the second one is Technology Park Basel. So these both can offer private and shared lab space, and the projects that are supported by us, we can offer free of charge access for a period of time. So here, just another word about our partners. So we are not only backed by our seven main partners, but also our domain partners that are also very happy to work with our companies in their given domain. It is also very important to mention that we appreciate that each project is different. So we'll really it's really important that our ventures really retain this full entrepreneurial freedom, and they can really decide what they want to do and how they want to do this. Of course, they also retain this full control over the extent to which our partners really become involved. So here about our portfolio companies. So in our first years, we supported 18 ventures. 8 of them already founded, 1 is sold, 1 stopped, and 3 are work in progress. And of course, we are working on adding a few more. Currently, of course, I would like to encourage all of you to go to our website if you would like to learn a little bit more about any of these companies. They have a very diverse scope. And our 8 financed companies collectively raised over $390 million from those top-tier investors here, with an average Series A financing of $28 million. So we are very happy with this number, and we wish all more successes to our old companies. And if you would like to become one of our portfolio companies, then I would like to encourage you to submit your application. Our next application deadline is 25th of April. You can find application form on our website. And also, if you have any questions or if you would like to discuss your project, then do not hesitate to reach out to us. Thank you.
Thank you, Martyna, for this excellent presentation. Our next presentation, we go alphabetically, is a company called Adoram. And Adoram is going to present their special therapeutic portfolio based on the antagonists of adenosine 2 receptor. Go ahead.
Hi, everyone. My name is David Peyoski, and I'm thrilled to have the chance to tell you about our project called Adoram. We're a seed stage pre-incorporation project based at the University of Geneva. Let me introduce you to our team. So you can see the management here on the top line. We have a shared passion for addressing unmet medical needs, and we're combining our diverse experience, myself in immunology, vaccines, and immunotherapy. I've worked in academia and industry and also been project managing for the past couple of years. We've been recently accepted into the InnoSwiss core coaching project to help move our lead candidates into the clinic. And joining me as the CSO will be Isham Ahmed. He has 15 years of experience in different disease models, including preclinical development. He's assisted delivery of 2 clinical drug trials and consulted for various companies. We're based in the lab of Leonardo Scapozza, a professor at the university here. His lab of about 30 people has a dedicated team that is capable of allosteric drug screening from hit to lead and everything in between. He's the co-founder of another startup, has a strong publication record, and is a board member of various societies. And I mentioned this platform to do allosteric drug discovery, and shown here are the team members who have worked very hard to get our results so far with the current project. So why would we want to block adenosine? It's produced by tumor cells and immune cells, and the extracellular adenosine in the tumor microenvironment binds to a whole range of immune cells, for example, NK cells, T cells, macrophages. And once it's bound to a certain pocket, it induces immunosuppression in all of these different immune cells, so they no longer attack the tumor. Therefore, if we can block this signal here, we would not only be able to improve a patient's own immune response, but also co-administered immunotherapies, for example, checkpoint inhibitor antibodies, CAR T cells, and cancer vaccines. In the clinic, there have been quite a few attempts to do this. You can see here we've grouped these companies as first-generation orthosteric antagonists because they have a few shared features. Several of these get into the brain, and that's not desirable. They were repurposed from other clinical indications to try and benefit cancer patients. And they're just not efficacious when there's a high concentration of adenosine. They weren't really designed to be able to withstand displacement from the binding pocket, which they have to compete with adenosine for. So there's a second generation. These ones are also in the clinic, and what has been coming out from their trials so far is that there's a few red flags for toxicity. We're waiting for more data. We propose a third-generation negative allosteric modulator, or NAM, and this is a unique molecular mode of action because it binds to an allosteric pocket in the receptor, and that then prevents adenosine from sending its signal into the immune cell. And once this NAM has bound there, we will show you data where we can restore the anti-tumor immune responses in different cell populations. So based on this, we can think of Adoram NAMs as a first-in-class because they are adenosine concentration independent, and the best-in-class because they have improved potency and safety profile over the competitors. Some data here showing our NAMs head-to-head with the orthosteric compounds. If you look here from lowest to highest adenosine concentration, these last 2 points over here, that's what kind of adenosine concentration would be in a tumor. And our molecules can inhibit the receptor regardless of these very high adenosine concentrations without having to increase the amount of our drugs. Whereas all these other orthosteric inhibitors, much higher doses would be needed to have the same type of receptor blockade. For example, 50 or 150 times more drug to achieve the same blockade. And if you want to see more of the biological data, that's all I have time for today, but feel free to set up a meeting with us. You can meet me and some of the other team members. We'd be happy to discuss and show you our roadmap and hopefully work together on this project in the future. Thanks for listening and have a great conference.
Thank you, David, for this elegant presentation. And we'll move on to our next presentation. In this case, Priyanka, she's going to tell you about Healiva. Hi, Priyanka.
Thanks a lot, Roy, and thank you, Thomas and organizer, for giving me this opportunity to present Healiva. And I'm delighted to present Healiva today. I'm the co-founder and CEO of Healiva. We founded Healiva in 2020, and we are located in Lugano, Switzerland. So what we are doing in Healiva. So Healiva, our mission is to personalize end-to-end chronic wound solution, making it more personalized and affordable. And we are combining, and I'm going to talk a little bit more about our technology, enzyme technology and cell therapy. Let's a little bit look into first the unmet need, what we have, and let's a little bit talk about the chronic wound patients. There are about 36 million chronic wound patients. And if you look into the numbers a little bit more, there are about 2 to 3% of elderly who have venous leg ulcers or diabetic foot ulcers. And about 25% of diabetic people have diabetic foot ulcers. Today, the therapies are not personalized, not affordable, and not effective. And overall, 60% of chronic wound patients remain unhealed. And if we look at where Healiva is present in Switzerland, it's about 3,000 patients still suffering from chronic wounds, and they never recover, and they need to get treatment all over throughout their lifetime. And we spend about 15.6 billion for the chronic wound right now globally. Little bit going into the cost. What we are spending, if we look into Europe, in Germany, we are spending about 6 billion annually. And in the US, we've spent about 25 billion. So in the end, we still have very high financial burden for the patient and society. Again, the patient repeatedly have to go through the surgeries. They need medical experts, and also they need frequent dressing changes and very lengthy hospital stays. What we are doing in Healiva, so in left-hand side, you have this different severity of wound and the different standard of care, the different treatments, what is presently there. And right-hand side, it's the different treatments, what we are offering in Healiva. So what we have from Healiva, from step 1, we have 4 different portfolio products, and we have products in every severity, for every severity of wound. So we start with enzyme. So we clean the wounds, we're making sure there is no infection, and it's painless, easy to use, and we give this treatment to all patient groups or all the chronic wound patients. Then we have a second product, which is prepared autologous blood patch from the patient blood itself, and it is prepared in the healthcare in the hospital setting. And then we have two portfolio products in cell therapy. We have one is an autologous cell therapy from patient back to patient, and we make a complete dermal layer, meaning we completely close the wound. And we have allogenic cell therapy, which is from healthy individual back to the patient. So we are covering complete spectrum of wound care, and our portfolio is comprised of enzyme and the cell therapy. Little bit going into timeline where we stand. Two of the portfolio is quite advanced. It's been technology transfer from third party, that's why it's quite advanced. And the first one, the autologous cell therapy, we will be launching in Switzerland next year. We are preparing our GMP sites, and we are also preparing to get it approved in Switzerland. For allogenic cell therapy, we will be entering into phase 3. Right now, we're currently working on the manufacturing process to scale it up. And the two other products is early stage, and this will be, of course, backing up our revenue for the later stage for 001 and 002. We are not doing this going to the market alone. We have partners at this stage which will be supporting us to bring these products to the market. We also have in collaboration with the university where we are doing initial studies, and we have support of the local cantonal level as well to support our innovation and expanding, helping us to expand our startup. We are tapping into more than 16 billion revenue because of, of course, the unmet need, and because of, the severity of the condition of the patient globally. So we have opportunity. We have very big opportunity to, of course, go into the venous leg ulcers and diabetic foot ulcers. There are still about 9.7 million venous leg ulcer patients worldwide, and about 10 million diabetic foot ulcer patients worldwide. Looking into a little bit of the team, of course, we have a very solid team of experts and KOLs who are specialized in wound care, enzyme technology, and cell therapy. Little bit going into the investment. We are opening our next round of investment, Series A, March onward, and why we are so strong for the investment for financial investors and strategic investors. Reason because we are tapping into an unmet need for the patient, especially for the chronic wound patient. And with our solution, what we are offering is we completely close the wounds. It's cost effective, and of course we are trying to rebalance the life of the patient, the work and the normal life of the patient, and they do not have to come back to the hospital after the cell therapy treatment. And why our complete portfolio is unique, because we are tapping into end-to-end wound care solution, starting from cleaning of the wound, making sure that they are completely healed. And two of our portfolio products is quite advanced, and one will start making revenue early on. So we're de-risking the revenue model, and we'll start the revenue process already next year. And we will enter the phase 3 trial with the 002 within 2 years. With this, I will end my presentation. And of course, if you like to personalize the wound care, make a difference, please reach out to me. I'm happy to answer any questions. Thanks a lot.
Thank you very much, Priyanka. That's a very clear presentation. I have one question for you. If I am a patient and I have a wound that requires attention, who will be contacting you, my physician or myself?
Good question, Rao. So we have a combination of the customers, you can say. It will be directly the hospitals whom we will be working together, okay, and the physician who will prescribe our therapy. So if it's an autologous cell therapy, it will be a second line of treatment, meaning the head-to-head with a smash graft. If they could not be cured with our allogenic treatment, they will be given the second line of treatment. So it will be the hospital who will prescribe our therapies. And what we are trying with autologous one, to get it approved by insurance company in Switzerland. It will be fully paid by insurance, and patient do not have to pay.
So you will be primarily convincing the physicians actually about the value of your product.
Correct. So we will convince the physician that it works. And of course, the patient will see the results of the treatment.
Okay, thank you very much. Okay, so thanks again, Priyanka, and we will move on to our next presentation on InnoSurge, and Thomas Roth will tell us what keeps him awake these days.
Thank you. I'm Thomas from InnoSurge AC. We are based in Aachen, Germany, and we enable AI-optimized surgical planning with just a few clicks. What is actually surgical planning and when is it needed? It's needed in many medical disciplines, and facial reconstructions are just one example. Let me illustrate that for you. This is Jack. Unfortunately, Jack has a bone tumor in his lower jaw, and this part of the bone must be removed. Afterwards, the resulting gap in the jaw must be closed and bridged again so that he's able to speak and chew again properly. And therefore, a transplant from the patient's own body is used. That's the gold standard. For example, a part of his lower leg. And since every patient, every case are unique, it requires patient-specific surgical planning to define this individual transplant design for this individual patient. And today, physicians are not able to do that on their own, and instead they assign external service providers who manually plan the transplant design. So how does that work? The physician sends the CT images to a service provider who then creates a 3D model. However, in the facial area, this is often a complicated process because metal inlays lead to artifacts and they have to be removed manually, and that can take several hours of work. Once this is done, the physician and the service provider meet up in a web meeting to discuss the requirements for this individual patient. For example, the lower jaw has to be removed by 70% and a fibular transplant is used. This is then executed by the service provider. And a couple of days later, they meet up again and discuss the results. However, this often leads to rework because of misunderstandings. But once everything is done, a cutting guide is 3D printed. That is used to cut the donor bone into segments that are then used as a transplant. Once the surgery is done, dental implants can be set and Jack can gain his quality of life back. However, this process takes up to 14 days and the physician has to put in up to 8 hours of work of his own time, even though it's an outsourced process. And because of all these problems, for all stakeholders, we found a solution. But let me present to you the problems for all stakeholders. For the physician, it's a time-consuming coordination that I mentioned and the low flexibility because the service providers only work from 9 to 5. For the hospital, outsourcing is expensive because they have to pay the service provider on top. And 8 hours of wasted time is basically 1 day of lost revenue. But most importantly, for the patient, we see a risk due to human error and a lack of optimization. InnoSurge provides the first automated all-in-one solution. The first step, it's a web application that's easy accessible and allows the physician himself to upload the CT images of the patient without patient data. We're using artificial intelligence for automated segmentation. So the physician gets a perfect 3D model within seconds. The second step is the virtual surgical planning, which is also a novel procedure that we developed, and we filed international patents for that. European patents recently were granted. The third step is the cutting guides again that are 3D printed. In the near future, this might be done by surgical robots. We are already preparing that. And the last step is a marketplace for patient-specific implants. Let's sum up our USP. For the physician, we offer a web application that's intuitive and easy to use. Physicians become flexible and independent and can do the surgical planning whenever they want and wherever they want. And it takes only up to 15 minutes, so it's up to 90% faster than today's solutions. For the hospital, we offer lower cost per planning case, approximately 3,000 euro per planning case, pay-per-use concept, and prices vary per country and application. But they also gain free capacities because they save so much time. And for the patient, we offer optimized results. But most importantly, we're not stopping with facial reconstructions. We build our software in a modular way. So year by year, we will add several other applications to our software suite. And this increases our total available market to almost 5 billion euro. InnoSurge's team is very interdisciplinary. It consists of software developers, engineers, and medical doctors. Now let's sum it up. We are InnoSurge. We turn manual into automated. We turn error-prone into optimized, and we turn several hours into 15 minutes. Now we are currently raising our seed financing round of 1.6 million euro, and we are looking for investors and angel investors. We already have several commitments and would like to close the financing round in the second quarter. Our key milestone is the market launch of our first application within the next 12 months. And there's a high potential because we will already be break even in 2024. And by 2026, our revenue is forecasted to be more than 30 million euro. Thank you for your attention and looking forward to hearing from you.
Thank you very much, Thomas. This really looks very exciting software. Does the physicians need any hands-on training when they acquire your software?
Well, we are building our software in a very intuitive way, so it's easy to use. They can't make a mistake because everything is guided step by step, guiding them through the process. However, we will of course offer training sessions, video tutorials, a guidebook, and they can also be supported with our stuff for the first 3 cases for free.
Okay, and my second question is, I mean, initially you are starting with the most difficult ones, I guess. Your customer base is very low, but as I see it, you are trying to expand so that you can go into multiple surgery indications.
Yeah, we are very strong connected in Germany and abroad as well, because we have this network of physicians that support us and that are interested in our solution. And that's why we have very strong confidence and won several prizes and are very well connected to pilot customers that give us early phase feedback already.
Okay, thank you very much, Thomas. Good luck to you. So our next presentation is Klavant. It is the cardiovascular medical technology, and we'll hear what are the exciting technologies they are going to offer.
Hello, I'm Michael. I'm one of the co-founders of Klavant, and today I want to present you IDA, the intraoperative digital aortography system. One problem in the conventional heart surgery is the manual intraoperative quality check. Some surgeons are doing it with eyeballing by hand, some are using a ruler or a caliper, and this leads to undetected leakages. And if you something don't see, you cannot correct it. This is why a non-precise information can lead to a wrong decision in the surgery procedure. This can lead to reoperation and revision, or unwanted escalations, and escalations can be mortality, morbidity, and quality of life. To solve this, our solution is developed to replace manual instrument to give surgeons a better decision-making and decreasing risk management. Our device is simulating the real blood pressure, and our advanced software can do reliable sizing of the aortic valve, checks the leakage and detecting them by stereoscopic and state-of-the-art measurement. Our system consists out of 2 parts. One is the imaging and pressuring part with 3D optical sensors, laser pattern, ultrasound sensor, and a liquid supply system. And the second is a control unit, which is needed for the imaging unit. Here takes place the imaging processing, image recognition, and the control unit is connected to a cloud service via API and the hospital IT infrastructure. Our intraoperative use cases are the aortic valve repair, the aorta ascendens resection, and the aortic valve replacement. We have around about 300 cases per year, and every case needs dedicated information to plan the next step during a surgery procedure, like visual inspection, tightness check, functionality, geometry of the aortic valves, and suture insufficiency check. The high surgical value will come from the use situation. A heart surgeon can use our device before the procedure starts and can adjust. For example, is it better to make a reconstruction of the aortic valve, or is it better to make a replacement? The surgeon can use it after he carried out a procedure to check the result. is everything fine? Are the valves tight again? Is the aortic insufficiency solved? Or can I proceed with the whole procedure? This will lead to better clinical outcome. And at the end, to less reoperations, less escalations, mortality and morbidity. The business we are addressing is quite huge. It's between 6 and 8.6 billion per year. Our revenue streams are single-use disposable sales, sales with a control unit for multiple use, and a monthly service fee we will charge. The difference in the pricing comes from the more competitive markets. Our status quo right now is we are ready to close a pre-seed financing round with 1.0 million. We raised already 700K to leverage 1.5 in public grants. And at the end of the year, we want to close a 2.5 million seed round. First revenues are expected in 2025, break even in 2026. And in 2030, we are planning to sell more than 23,000 disposables. We ended with in vitro trials and preparing the first in human, followed by prototyping and go to market. If you are ready to become part of a heart surgery game changer startup, we are happy to answer all your questions. Come back to us. Thank you for your attention.
Thank you. We move on to our next presentation, which is oncology, the therapeutic products. So it is presented by Thomas Merling, the CEO. Thomas, could you please tell us about the exciting opportunities that you are trying to pursue?
Yes, thank you very much for the opportunity and welcome to Laevoroc Oncology. My name is Thomas. I'm a hemato-oncologist from Frankfurt, Germany, and we have founded Laevoroc Oncology in 2019 with a focus on immuno-oncology. Our mission is to develop new second generation or game-changing immunotherapies to treat cancer. Now, we are running 2 programs in immuno-oncology. Our first one, let's look at that one, is LR01. LR01 is a T-cell engager construct, which is a phenomenal new technology to activate the immune system, mainly T-cell lymphocytes, to recognize tumor cells and basically destroy them. The way they do that is that we have bifunctional antibodies that at one side bind to the T-cell and at the other one to the tumor cell. This technology has already achieved substantial and game-changing advances in other leukemia types, like acute lymphoblastic leukemia, and most also recently in multiple myeloma, a different type of a blood cancer. Now, this one here is for acute myeloid leukemia. Now, let's talk a little bit about acute myeloid leukemia. This type of leukemia is the most often diagnosed leukemia type in adult patients. We see about 125,000 cases diagnosed every year. But this type of leukemia is not really treated very well. The 5-year overall survival stands at 29% in the United States, and it may be even less in other territories of the planet. The disease is basically incurable, except that patients are able to get a bone marrow transplant from a related or unrelated donor. It is only that some of the subgroups of AML, smaller ones, benefit from small molecules that have been introduced to treat FLT3 mutations or IDH mutations. But the broad population is still treated with chemotherapy. Now, we think our construct has the potential to revolutionize the therapy for AML. There are other constructs in development, but we think we differentiate at least in 3 ways. The first thing is the target. We are targeting FLT3, which is expressed on about 95% of all AML. The second advantage is that current constructs have to be given in a continuous infusion, and patients have to carry an infusion pump all day. Our therapy can be given once a week. And the third one is that our technology we're using for this construct is already in 4 clinical programs, so we know about the excellent safety profile of our construct. So we're very excited about this technology, And this is a technology we would like to license. Now, our second program is also for leukemia, among other utilities. But this compound, which is able to activate the innate immune system, the immune system that protects us from any invader that is foreign to our body, that happens, for example, in virus infections or when we get COVID vaccination. This is recognized as foreign. The immune system presents this to the immune cells, and those are creating lasting immunity. Now, we have chosen for this metabolic immune checkpoint agent an indication that also suffers from a huge unmet need. This is leukemia relapse after a bone marrow transplantation. These patients have the potential for a cure, as I told you, but actually still 50% of these patients are relapsing and carry a very bad prognosis. Overall survival is between 3 and 16 months, and there is no real standard of care available. Our therapy, we know that already, is activating the transplanted immune system and is potentially eradicating the leukemia that's coming back. And this is a major difference to what's currently achievable in this disease. We are very passionate about this program and would love to bring this to patients. Now, this is what we would like to achieve with this 30 million funding in a Series A that we're looking for. So we would like to license the T-cell engager technology and take it to the first inpatient clinical trial until 2024. In the meantime, we would like to take the LR09 program in patients relapsing after stem cell transplantation to first in human clinical trials already next year. So all of these advances will be achievable according to our financial plans until 2024, and you will see that most of the money will go into the LR01 program to achieve that goal of first in human trials, but we will have already completed phase 1 dose escalation studies for LR09 with that kind of funding. We also will need to spend some money on the development of the company until then, and this is making up the total investment we're currently asking for. So many thanks for your attention, and very happy to take any questions you may have.
Yeah, thank you very much, Thomas. I have one question for you. How would you like to produce the antibody that you are referring to?
Yeah, this antibody has actually a quite simple manufacturing process. It's used in conventional CHO cells, and it follows the general principles of manufacturing antibody. Despite this antibody is fragmented, it's not a full IgG antibody.
Okay, thank you very much.
You're welcome.
I wish you good luck.
Thank you.
Thank you. Okay, we'll move on to the next presentation, Ramino Bio, by Dorit Cohen-Carmen, Chief Scientific Officer. Go ahead, Dorit.
Hello, everybody, and thank you for the opportunity to invite me. And today, I will take the opportunity to discuss about Ramino biotechnology, which we would like to enhance branched-chain amino acid catabolism for treating maple syrup urine disease. You see this? So this milk is actually categorized as protein-containing food. Most of us consume proteins in our daily diet, but for some, eating protein is life-threatening. We at Ramino Bio are developing a drug to treat maple syrup urine disease patients that cannot consume proteins. Currently, there is no therapy for those patients. So at Ramino Bio, our mission is to lower branched-chain amino acids, which are actually 2 built-in blocks which are elevated at maple syrup urine disease in other chronic cardiometabolic diseases such as type 2 diabetes and more. The mechanism of action and the target, we would like to lower the elevated branched-chain amino acids by accelerating their breakdown, by inhibiting with a small molecule compound the inhibitor of the catabolizing complex. We were established 2.5 years ago, and the technology origin is from Professor Libin Wang from UCLA and Professor David Chang from Southwestern University. Both studied those type of molecular pathways. We are funded founded by OrbiMed, Takeda, and Johnson & Johnson, and we are part of the Futurix Biotech incubator in Israel. The indications that we are aiming to treat is proof of concept maple syrup urine disease, and also the cardiometabolic diseases, type 2 diabetes and heart failure. What we have decided and succeeded to achieve so far, we have today 2 novel and potent new chemical entities with good druggability properties. The PCT was filed already. With those compounds, we were able to show ex vivo efficacy studies in maple syrup urine disease patient cells and in vivo proof of concept in mice. And we are now in a stage of hit to lead and optimization stages. So, few words about branched-chain amino acids and maple syrup urine disease. This is a rare genetic metabolic disorder in which the body cannot break down the branched-chain amino acids, and they're accumulating in the body. I should mention that BCAAs, branched-chain amino acids, are 3 out of the 9 essential amino acids that the body cannot synthesize by itself and need to be consumed from the outside. The patients, if those patients are not given a specific BCAA diet, it is life-threatening, and there are a lot of neurological damage because the byproducts are toxic to the body and to different tissues. This is a genetic indication, and actually the catabolizing complex, as you see here, has mutations that leads to either a full loss of function or partial loss of function. So if the catabolizing complex is disrupted, there is accumulating branched-chain amino acids in the circulation and tissues. The current treatments have a lot of limitations. The patient should be on specific diet control. They have day-to-day monitor of their blood and urine for elevated BCAAs. They have episodes of sometimes psychiatric bursts, kidney failure, ADHD, and other complications with a lot of hospitalizations and constant monitoring of their blood and urine. For severe cases, there are sometimes liver transplantation, but as you know, this has limited availability, high risk, and sometimes a lot of failure. We at Ramino Bio aiming to answer this need for maple syrup urine disease patients by enhancing the BCAA breakdowns. So with our technology, how a maple syrup urine disease and cardiometabolic patients can benefit. The drug that we are developing for lowering BCAA in tissue and circulation will reduce brain damage and life-threatening episodes, will enable higher consumption of BCAA needed for protein synthesis, for muscle and bone buildup. Actually, as you know, for protein synthesis, you need all the essential amino acids. And if you don't have them, how you can build actually bone and muscle. It will enable to lower accompanied risks such as depression, ADHD, kidney failure, and related hospitalizations, and of course, a higher life quality for the patients and their families. Lowering of BCAAs levels via our technology can be applicable also to patients with insulin resistance, type 2 diabetes, heart failure, which will improve cardiac muscle contraction, adipose and other tissue redox homeostasis, mitochondrial function, and glycemia. It is very known mechanism for elevated branched-chain amino acids that are taking part in propagation of insulin resistance and type 2 diabetes. Where we are now, so we started in the tech transfer, hit identification and selection, hit optimization. We are now in vivo proof of, we already proved the in vivo proof of concept with an optimized hit, and we are about to have a lead selection by mid-2023, and this will be the end of funding. Hopefully by the end of 2024 with IND submission. As for the achievement, we have 2 novel compounds that were shown ex vivo efficacy studies in many maple syrup urine disease and in vivo proof of concept in mice. The founding team, the technology is based on novel findings by Professor David Chung and Professor Weibing Wang. I'm Dorit, a biologist by training, got my PhD from the Weizmann Institute of Science for Molecular Virology and Metabolism, and I did my postdoc for Huntington disease. After more than 15 years in the academy, I have moved to the industry to be part of the effort to develop therapies for human diseases. Dalik, our principal scientist, medicinal chemistry, has more than 20 years experience in biotech industry for small molecule compounds development. And we have two scientists, expert scientists with a lot of knowledge and experience in vivo, in vitro studies. So why Ramino Bio? We are now looking for fundraising or collaboration. Ramino Bio's technology is a disease-modifying mechanism. It's a maple syrup urine disease management, rigorous diet, and a significant unmet need. Patient selection based on our in-house ex vivo testing using the patient cells is possible. The novel compounds showing efficacy and proprietary knowledge. The team with the in-house expertise in medicinal chemistry and the contact with Professor David Chung. The biomarkers are clear, quantitative, non-invasive, and the clinical proof of concept can lead to additional metabolic indication. We are interested to connect with you for collaboration or funding till phase 1. Thank you and good luck.
Thank you, Dorit. This is a very new disease for me. I have two questions. One, how many patients actually exist with MSUD?
Okay, since this is a rare genetic metabolic disease, there are about 2,000 in the US and 4,000 at Europe. Okay. But there are other communities around the world, in Asia, in the Mediterranean, in Mexico, in South Africa, with the founder mutation of this disease. Yeah. As I said, we are aiming to treat maple syrup urine disease, but this drug can be a platform for other cardiometabolic indications.
Okay. And the second question I have is, so you will develop a small molecule that's orally active and in a circulatory system decrease the concentration of these branched amino acids?
Exactly.
Okay. Thank you very much.
Thank you.
Appreciate it. Okay, our next presentation is Testasy.
Hi everyone, I'm Jayla and I'm here presenting Testasy. 70% of medical diagnoses are based on lab tests, and 70% of deaths are due to preventable chronic diseases. However, lab testing has not left your GP's office in the last century, and in most countries, you still need your GP to tell you what tests you need and when you need them. Average cost of all the lab tests is approximately 500 francs on average. Your regular lab testing journey looks like this. It involves multiple steps, going to your GP, talking to your GP, and receiving some basic recommendation based on this report, which is several pages long, involves all the medical terminology you have no idea about, alongside numbers that don't really tell you anything. So naturally, you need your GP to decrypt this, and based on that, he would give you some basic recommendation about your health. Testasy wants to streamline your medical journey. We want to make testing accessible, so you decide what tests you need and when you need it, giving you control over your health. We want to make it convenient, so instead of you going to the test or to the lab, the test comes to you. We want to make it consumer-driven. By cutting out the GPs, hospitals, and everybody else, we act as a middleman, giving you health insights that you need and understand. This is thanks to Testasy being comprised of several components in its platform. Number one is Testasy lab tests, which involves everything from blood to STD testing to fertility and viruses. It also involves telemedically assisted self-sampling with healthcare professionals only. Number two is our partner services, established businesses offering a range of health services such as mental health, telemedicine, and second opinions. And on top of that comes the health insights, a comprehensive insight related to your personal health in a context. It shows you easy to understand results and actionable data aggregated from the two previous components. This is what it looks like to you, easy to understand results. You understand where you are in terms of your health and what's wrong or what's normal. It gives you information so that you can have informed discussion with your doctor, as well as explore potential next steps. And this can be applied to a range of lab tests, from blood, urine, sperm, genetics, and viruses, as well as use cases are limitless, from general population to professional athletes to corporate clients, you name it. We actually tested this model successfully during the pandemic, as in 2021, we launched Testasy to offer telemedical testing solution for COVID-19, where you could perform a test remotely under supervision and receive a certificate with QR codes valid for travel globally. That saved you hours of waiting and efforts. And we opened in Switzerland, Poland, Estonia, and currently also opening operations in Thailand. We had action-packed 8 months of existence. We launched in Warsaw in April, then in Switzerland. Then we quickly became the only telemedical partner to Abbott Pharmaceuticals in the EU. We went on to introduce saliva PCRs in Switzerland as well as in Poland. We were the first ones to do so. And we're working on at-home blood collection devices alongside other Swiss startups. So at-home testing industry is booming. It's destined to be a $2 billion industry by 2025 as people realize that most things can be done from home. It's also estimated that increase in use of telemedicine went up almost 40-fold as people are becoming more accepting of telemedicine on both sides. Obviously, there's lots of startups successfully operating either as telemedical startups or at-home testing, but none of them actually offers the two. Testasy hits the sweet spot. What we do is we ship you the test, but instead of just giving you instructions, we also hold your hand, make sure you sample correctly, make sure you sample enough, make sure you is you and that you receive the help and that you're not afraid to do any types of tests. Why us? We have already established network, labs, commercial partners, corporate clients, all of which would love to collaborate with us. We have a proven concept wherein we worked on COVID-19 with rapidly changing regulation and temporal pressure. We also have proprietary technology, software that is significantly streamlining all processes related to testing, and that has been adopted by a large insurance company in Switzerland. Our medical team is there, our business team is there, as well as very good support from our tech team. If you need any information about investment or financials, please contact me directly. Here's my email and here's my mobile. You can call me or WhatsApp me, and I would be happy to address your questions. Thank you very much.
Thank you. We are getting to the end of the presentation, and Catherine will tell us about Ymmunobio.
So it's a great pleasure to talk about Ymmunobio today. We are a preclinical stage Swiss biotech company, and we are developing CEACAM1 antibodies for the treatment of different cancers. And in the next 5 minutes, I will take you how we make cells talk. I'm Catherine Ropalla. I'm actually the founder and CEO of Ymmunobio. I have about 25 years of experience in drug development. My main career I spent at Roche, Celgene, and Bristol Myers Squibb. And also at Bristol Myers Squibb, I spent 8 years in the development of anti-PD-1s. So very familiar with the therapeutic area and the drug development process. And I'm supported by Dr. Bernhard Singer. He is the inventor and the chief scientific officer. He has over 25 years of experience in the research field of CEACAM. He has spent most of his career at the University of Freiburg, the Charité Karolinska Institute, and the University of Essen, and he is by training an immunologist. We are supported by a team of advisors. Professor Iris Helfrich actually also is our link to one of the top KOL melanoma treatment centers in Europe, which is Dirk Schadendorf's clinic in Essen. She is also helping us with the in vivo experiments, so one of top experts on the preclinical side. We have Keith Skubitz, who's professor of medicine and hematology oncology at the University of Minnesota, treating clinician and also CEACAM expert. And then we also have Bruno Osterwalder, a very seasoned drug development expert. In a nutshell, I also want to give you an overview of the company. First, we are developing very differentiated CEACAM1 antibodies. They are differentiated from the very few CEACAM antibodies that are currently in preclinical or clinical development. Our antibodies have dual action. They have a direct co-stimulatory effect on immune cells, which is a unique feature, also very differentiating from normal checkpoint inhibitors. But on top of the co-stimulatory effect on immune cells, we also have a checkpoint blockade. So we have a dual mechanism of action. And our pipeline consists of 3 generations of CEACAM1 antibodies, second generation YB200, 300, the third generation, and YB400, the first bispecific CEACAM1 antibody. We have robust scientific data, both in vitro and in vivo activity. Clearly, multiple experiments also having been published in high impact journals like Nature, showing the expansion, maturation, and prolongation of survival of our antibody on immune cells. And also we have first in vivo activity data in PD-1 resistant tumors in a preliminary mouse melanoma model. And I believe this is maybe the most exciting piece of information, is that nobody has seen activity of an immune targeted agent in a PD-1 resistant melanoma model with B10F16 melanoma cell line. So this is really a very encouraging result for us. We address high unmet medical need. With the dual action, we actually believe we can overcome PD-1 resistance, also shown by the preliminary in vivo data. We have this existing collaboration with the top melanoma center in Europe, Dirk Schadendorf's lab. We have this existing collaboration. Melanoma, despite major advances with the checkpoint inhibitors, of which I have been part of the journey, is only 40 to 45% of patients respond to checkpoint inhibitors in melanoma. And unfortunately, more than 50% of these patients who respond to checkpoint inhibitors progress within 12 months. It's still the 5th most common cancer in the US. And as I said, we are working with a top melanoma research group and have access to one of the largest melanoma patient data or patient sample banks. And the mechanism of CEACAM1 is actually, while we focus on the lead indication melanoma, can be applied to multiple tumors, and I come back to that in a minute. Also, the development of the CEACAM antibodies is underpinned by a strong IP protection and the IP composition of matter and method of use patent until 2040. The rationale for developing anti-CEACAM antibodies in immune oncology is actually based on 2 pillars. The first pillar is actually really the physiological activity of CEACAM1. It's expressed on immune cells and is actually a perfect example of a cell-cell communication molecule, CEACAM1. And that's why we also make immune cells talk. They are membrane anchored and they influence directly the growth, the apoptosis, and the activation of immune cells. And then our in vitro data have shown beautifully how B cells, T cells, and antigen presenting cells actually are being activated by our antibody. But also CEACAM has a pathophysiological role. It's actually dysregulated in cancers. It's part of the carcinoembryonic antigen family. It's neo-expressed in several cancers, such as melanoma, lung cancer, pancreatic cancer, and is associated with unfavorable prognosis. And that leads me actually to the main differentiation of our CEACAM antibodies, of which YB200 is the lead compound. We have this direct co-stimulatory effect, which is mimicking the physiological activity of CEACAM1. It's directly activating immune cells, leads to prolonged survival of T and B cells. It leads to maturation and differentiation of these cells, which then help, targeting the tumor in cells and drive tumor cells into apoptosis. That's the main mechanism of action we believe that will be successful in overcoming checkpoint inhibitor resistance, because we have a direct effect of stimulating the immune cells and can potentially turn cold tumors into hot tumors for immune therapies. On top, we are interrupting the checkpoint blockade that is happening between the tumor cells and the immune cells. That's in the pathophysiological state of a tumor, where you have this checkpoint inhibition, and the CEACAM1 antibody will be able to disrupt this checkpoint inhibition by the tumor cells and CEACAM1. That's the main differentiating factor. And if we come to a conclusion here, it's we have a robust scientific rationale and also robust scientific data already. There's a well-established role of CEACAM1 in the immune system as a cell-cell communicator, and plays a major role in tumor progression. The two lead compounds have shown to lead to expansion, maturation, and prolonged survival of immune cells. These data have been published in high-impact journals already. YB200 has already shown activity in PD-1-resistant melanoma models in vivo. And actually what we are looking for is, we are seeking seed funding. The company was founded about 5 months ago. We are looking for seed funding of 1 million Swiss francs. In the next 9 to 12 months, what the $1 million CHF would allow us is to generate key data, reach the first milestone of high quality proof of principle data in vivo, and also to de-risk development, so it enables us for Series E funding. This would include confirmatory in vivo study, preliminary CMC development, such as humanization of the two antibodies, and exploratory safety studies. And the goal would be an IND submission for first in human study for YB200 early 2024. And with that, I would like to thank you very much for the opportunity to present.
Thank you very much, Catherine. That's a very exciting story with respect to immuno-oncology and development of the resistance with the first generation molecules. We need every facet of the co-stimulation as well as checkpoint inhibition to continue to treat the patients for better outcomes. And I really believe that you will have exciting people chasing you for your next generation immunotherapy cancer. I'm just surprised that you are trying to raise so minimum money, but I wish you good luck in every sense.
Thank you. Thank you.
Yeah. Okay. Well, this is the end of the session of the Rising Stars, and I'm really glad that you all took time to present at the SACS conference today. And thank you very much, Thomas, for organizing this meeting very effectively. And then I hope that you will reach the appropriate audience that you are targeting and be able to raise sufficient funds to make your dreams come true. Because tomorrow's companies are today's seed companies, and today's rising stars are tomorrow's professional, successful drug developers. So I wish you all good luck, and thank you again and for participating in this conference.